Thanks everyone for joining. It's my pleasure to welcome Katie Chang, who's going to be joining us today. Katie is currently an assistant professor of electrical and computer engineering, computer science, and biomedical engineering at Vanderbilt. She received her PhD from Stanford and was a postdoc at the NIH Intramural Research Program. She currently leads a lab called the Neuroimaging and Brain Dynamics Lab, and she's focused on advancing our understanding of human brain function through techniques for analyzing and interpreting neuroimaging data. So we're excited to hear. Today she's going to be talking about functional MRI of dynamic neural and physiological states. So without further ado, I will let you take it from here, Katie.
Great. Thank you so much for the kind intro, and thanks to you and Armin for organizing this talk series. So yeah, I'm going to talk here about some of our recent studies on fMRI dynamics. Specifically focusing on these neural and physiological state changes, and happy to clarify anything or discuss anything anytime. So just to set up a bit of overall motivation, with fMRI data, we're getting this four-dimensional picture showing how neural activity is dynamically changing across the whole brain. And traditionally, there's been a large focus on spatial mapping in fMRI. So which areas are activated in certain conditions, or what are the networks of regions that show high correlation to each other? For example, what are the functional boundaries between different regions in the brain? But at the same time, there's been growing and complementary interest in mining the temporal or spatiotemporal information in our data. And so in that context, some of the questions that our lab and others have been asking are, how do brain networks change their interactions over time? Whether the temporal properties of our data may indicate differences between people and provide useful biomarkers, as well as how we can perhaps capture and model real-time changes in someone's brain state, such as how awake or drowsy you are, or emotions you may be experiencing from one moment to the next. So I'm going to briefly share first a couple examples of some work on linking temporal features of fMRI to individual differences and clinical traits. Then I'll discuss some work on decoding ongoing changes in alertness from fMRI, interactions between alertness and physiology, as well as how we can reconstruct continuous respiratory and cardiac information based on fMRI. So there are many methods that have been proposed over the years for parsing fMRI data into meaningful features. And one of the approaches shown here is called coactivation patterns, or CAPS, that was proposed by Xiao Liu. And so as depicted earlier, we can consider our fMRI scan to be like a movie where we're observing a sequence of activity patterns at each TR. And we can call those co-activation patterns, since the areas where the signals are high at the same time are essentially co-activating. And so then to describe the structure of these patterns, what was proposed in this study was to assign each TR to one of several clusters called the state, whose pattern is represented by the cluster centroid. And so then having turned our fMRI scan essentially into the sequence of states, we can look at metrics such as the amount of time a person is within a certain state in a scan, or frequency of switching between different states, and related measures, and try to relate those to phenotypic measures. And so this is one recent collaboration led by Lucina Udin's lab, where they related transient fMRI coactivation patterns to depressive symptoms based on neuropsychological assessments, using this large sample of participants from the NKI dataset. And so this analysis indicated that there are several different coactivation patterns, including a state mainly localized to default mode network. And this was found to be more frequently expressed in individuals that had higher levels of depressive symptoms, which is perhaps consistent with other work that's implicated the default mode network in internal and self-referential thinking. Whereas another one involving more of the cognitive control network showed more of an opposite trend, actually, whose occurrence was inversely correlated with depressive symptoms. This study was actually somewhat, the findings anyway, were somewhat consistent with another study that I had a chance to collaborate on when I was a PhD student, led by Paul Hamilton, which looked at depressive rumination. And so, briefly, for the fMRI analysis, we were looking at how long a subject's brain would dwell in a state where there was higher activity in the default mode network compared to a task positive network, which comprised executive control and salience networks. And we found that the longer they spent in a default mode dominant state, the more they showed a tendency toward a depressive or maladaptive form of rumination. That was gauged by questions like how much they might be thinking about their faults or mistakes. And by contrast, the less they showed that default mode network dominance, the more they had a tendency toward a more adaptive or constructive rumination. So indeed, it's been an exciting area of fMRI research to try and focus on discovering different features of brain activity that relate to clinical traits, behavioral traits. And this can not only be used for giving us clinical biomarkers, but also for understanding the neural basis of individual differences. And one of the main approaches for looking at individual differences with fMRI has been based on connectomic information, like structural and functional connectivity from different brain regions. And so, but there's at the same time been a largely separate line of work on the dynamics of fMRI time series measured from local regions, like the variability of the bold signal, showing that this may also carry interesting information. And so something that my PhD student, Sheng Chao, came up with and decided to investigate is whether certain features in the local temporal activity across different regions also carry information about individual differences. And so using resting state data, if we extract and we look at the time courses of different brain areas, different atlas regions, we might notice that this is a cartoon actually because it's quite exaggerated. This is not real data, but you can often see that there's a difference in the spectral characteristics of regions. And so you can imagine kind of collecting that information across different brain areas and coming up with a vector that essentially summarizes that scan in terms of that temporal feature, the profile across those different brain regions. And so once you kind of characterize the data in this way, we can ask questions like to what extent that kind of profile is stable within a person, maybe just is repeatable within that person across different time windows or time or different scans, whether it's distinct between individuals, and whether it has any relation to behavioral and cognitive traits that were measured outside the scanner in a different session. And so just a few more details about the study. We used the Human Connectome Project data and a 90-region atlas and considered a measure of the power spectrum based on the fractional amplitude of fluctuations below 0.1 hertz, which has been called FALF in the literature, and a complementary measure of temporal complexity based on sample entropy. And so one way that we can start to test if these temporal profiles carry information is to follow something like a fingerprinting approach proposed by Emily Finn. So since each of these connectome subjects was scanned over 2 different days, we can ask if the spectral profile from a subject on one day has the highest match with themselves on another day compared to with any other subjects. And so we actually did find that the proportion of subjects who matched best to themselves between those days was around 90%, a little less than 90%. And so then we also wondered how much this depends on the spatial regions of the atlas we choose, because does it actually make a difference where those regions are that we're sampling? And we didn't do extensive comparisons, so I think that this is an area that would be interesting to look into further. But one thing we did do was compare this identification rate of the 90 region atlas with one that was based on 14 networks that those regions were a part of. So if we average things more coarsely and put them into 14 features, how much does it do, or how much does the performance change? There are less features, of course, but it doesn't seem to identify quite as well. And so in a way that's kind of comforting, because if anything identifies, then we have to think about what that means. But I think there is definitely more work to be done. And so one last thing here is that we also explored whether the self and complexity features could potentially co-vary with a subspace of behavioral measures collected from these subjects, and we implemented that using canonical correlation analysis. And using permutation testing, we found that there was a significant moat that loaded in the following way onto different behavioral features that we had pre-selected before the analysis. And I think there's always some trickiness in if we can exactly interpret these linear weights. But we did find that measures like sleep quality and sustained attention ended up as some of the strongest weights, which is interesting because previous work has shown relationships between frequencies in the bold signal and states of drowsiness. And that's something I'll be talking about a little more later on. And so overall, this work is kind of an exploratory analysis, and I think I was just intrigued that it seemed to kind of work, but I think there's definitely a lot more to dig into. And also to kind of reconcile what might be stable changes we see in temporal dynamics, or stable aspects of temporal dynamics within a person, to actually more like the moment-to-moment state-related variability, which can also change characteristics of fMRI data. So the studies that I just showed indeed tried to relate fMRI dynamics to differences between individuals or to these stable behavioral and cognitive traits. And those measures are often collected outside the scanner. But we could also ask to what degree an fMRI measure also really captures the more instantaneous mental and physiological states that people are going through, and there's support for this as well. So considering this early study, for example, it was found that the amplitude of spontaneous fMRI signals in the motor cortex measured before a stimulus came on was related to that person's behavioral response, in particular, like the force of the button press that they made. And here, the topological organization of connectivity that was present before a faint stimulus came in was indicative of whether the person ended up perceiving that stimulus or not. There have been a wide range of different states that fMRI dynamics have now been linked to, including attention, mind wandering, and emotion. And this is a review article led by Caroline Martin, in which she synthesized a lot of this recent literature. So, so on the other hand, I mean, there is, it's potentially kind of a confound if we, are trying to find a stable measure of brain function or traits, based on fMRI, if it is the case that these signals are kind of reconfiguring depending on what state a person is in, and particularly what state they're in when we're scanning them. But on the other hand, it also is giving us these changes that we observe are also giving us a window into how brain activity may be dynamically changing to support these states. So the study of these dynamic brain states then may be interesting in and of themselves. And so in that context, one area that we've been particularly interested in is vigilant states. So I'm going to use this term to describe the range of functional states between alert wakefulness and drowsiness and light sleep. So I know that there may be slightly different uses of vigilance in the literature. And one reason that this is relevant for fMRI is that people do get sleepy during scans. It's been shown that people can fall asleep within just a few minutes. However, it's also important because fMRI signals do change quite dramatically as people become drowsy and fall asleep. So there can be systematic changes in the amplitude of signal fluctuation as well as the connectivity across the brain. And several studies have also shown that the connectivity between specific brain networks is potentially modulated as a function of vigilance. And so, just as one example from one of our studies, uh, here we had monitored EEG simultaneously with fMRI, since EEG provides a more, I guess, well-validated measure of vigilance. You can look at things like the relative power in frequency bands such as alpha and theta. And we're looking at whether changes over time in these EEG measures of vigilance were related to changes in fMRI connectivity over time. And the data seemed to indicate that when a person was in a state characterized by lower alertness, there was stronger coherence overall between the networks of interest, default mode network and attentional network, compared to when the subject was in a likely higher alertness state where you can see more negative correlations. Um, and so one thing that this suggested is that some of the dynamic changes that we see in fMRI signals across the course of a scan may relate to the ongoing brain states of that person. And it also led us to ask whether, to what extent it could be possible to use fMRI itself to try to come up with a measure of the moment to moment like change in a person's vigilance state if we didn't have EEG recorded, which is often the case for our scans. And several studies have done something like this in terms of trying to stage sleep, for example, work by Tagliazucci and colleagues. And so the idea here was to kind of extend this work in the direction of trying to infer continuous levels of alertness when people are more at the awake end of the spectrum. And so the overall idea is, can we take in an fMRI data set as input and output essentially a full time course that captures a person's level of alertness from that fMRI data alone, which would then match well a measure collected in EEG if that was indeed available. And so the basic approach here was based on spatial pattern information. And so if we take concurrent EEG and fMRI data, we first learn a pattern of activity related to vigilance by looking at the correspondence between the two and coming up with a spatial pattern. And then if we're given a new fMRI data set that doesn't have EEG, the idea is that we can use this pattern and project it onto the data set in order to derive an estimated level of alertness at each successive fMRI volume. Here is in this example shown in red. And then what we did was to validate it, we compared it to EEG that had been measured in that scan. But this was only for the validation. It wasn't used to derive that index. And so we kind of separated our data into the training and testing sets in order to be able to do that learning and evaluation. And so this was when, in this study, we had mainly shown, this was actually in monkeys, we had shown that it was possible to derive a continuous fMRI index that matches to a fairly good extent the electrophysiological signals that were being recorded in the monkeys. And in a subsequent work, we wanted to try and do this also in humans, and also to be able to capture, see if this captures not only variations in the brain's electrical activity, but something relevant to behavioral variability. So can we actually use this to perhaps parse some of the variability in behavioral performance that could happen during a task? And so here we used a task that presented intermittent auditory tones, and subjects were asked to make a button press response as soon as possible. And even though it's quite a simple task, people can actually be pretty variable in their performance. So sometimes they may like respond quickly, and sometimes they will maybe miss it completely, or take a longer time to respond. And so the analysis we did was to basically isolate a brief window of time before the stimuli came on and calculate that estimated alertness level, and then see if that could be potentially used to predict the upcoming response time, or whether or not they hit or missed the target. This work was led by Sarah Goodale, a PhD student in our lab. And in terms of the hits versus misses, one of the findings was that indeed there seemed to be a separation of the fMRI alertness index in terms of when people would successfully respond to a stimulus versus when they missed it. And we can also see differences in the time course of this alertness index. So if we were to zoom in around the time zero being when the stimulus was presented, and look at that how it looks in the average when subjects had made a response versus when they didn't, those also differentiated. And confirming this analysis, it also seemed to differentiate before the stimulus was presented, as well as seeming to cause some transient increase or having some transient increase afterwards. And we speculated that this might be actually due to the tone waking people up sometimes, because it was a little bit, it was presented very intermittently. And so as an additional check that this may be reflecting vigilance, we had EEG recorded as well in these task scans. And this too indicated that subjects seem to be, if we look at the power spectrum of the EEG, the subjects were, that they indeed seem to be more wakeful before trials that had responses compared to trials that didn't, where you see more low frequency power. So I think that we were excited that these results suggest we may be able to infer at a relatively high temporal resolution these arousal states that are linked with behavior based on fMRI alone. And so to dig into this a little further, we then, instead of only considering the hits and misses, we took the trials where people had been responding and wanted to see, can we tell a bit how whether the person makes a fast or a slow response, and also found a higher level of estimated alertness in trials where people responded more quickly than more slowly. And then across the group, so this is within individual measure, but when we pool different subjects and we divide their reaction times into these fast, medium, slow responses or misses responses, we also see this effect. Just taking the time. Okay, so the last thing we did here was it seems that we can estimate vigilance from from the whole brain patterns, which is used in the results I just mentioned. But what if we try to pare that down to just a very small number of fMRI voxels? And so here we experimented with whether we could successively reduce the number of positive and negative intensity voxels to their top, up to 1%. And then we found that particularly when we maintained voxels with both positive and negative polarity in our map, we were able to continue achieving relatively high performance. And that was in contrast to if we only selected subsets of the highest intensity voxels with only the negative or only the positive polarity. And so I think that makes a lot of sense, because these very widespread changes in fMRI signals that correlate with vigilance are somewhat redundant with each other. They're quite correlated across the brain. And so we can kind of compress that by essentially selecting regions that have this highest correlation. But then the information between the positive and negative voxels seemed to bring further information that was specifically relevant to doing this alertness prediction. And so in terms of which regions showed this kind of effect, interestingly, the visual cortex popped out. I think, to one extent, I wasn't super surprised, because we often see very strong signals in the visual cortex and across many fMRI tasks. But also there had been related work showing that that vigilance or changes in vigilance or arousal during tasks came with correlation or came with activity in visual cortex. And we also find ventricle signals here, and also we're a little surprised at first, but actually this is consistent with other studies as well. showing that that in sleep, but potentially also in wake, there are slow global fluctuations in the brain's vasculature, which may modulate as well the flow of CSF in the brain. Um, and so, given that we see very widespread changes in brain hemodynamics that correlate with these fluctuations in arousal, that this might also be occurring in what we're seeing here. And so we think that the patterns that we're picking up on might have some relation not only to the neural components of alertness, but also to the brain physiology that's happening at the same time, the autonomic activity. And so just to expand a little bit further on that, when there's an increase in alertness, this comes together with changes in our physiology, like an increase in heart rate or constriction of blood vessels relating to a sympathetic nervous system response. Um, that that change in vascular tone can be measured in the periphery as well. And so if you have PPG signal and you look at amplitude changes, you can see sometimes that there are these drops in the amplitude of the pulse waves, which correspond to changes in blood volume. And so these studies were led by Jeff Dunn's lab, and we saw that these autonomic events measured in the fingertip were tightly linked with global changes in fMRI signals that resembled patterns that we had seen correlated with arousal. And so perhaps this autonomic response that's happening together with changes in arousal comes with a constriction of blood vessels in the brain as well, which may drive some of these changes. And so we think that overall, that pattern we are picking up on, and a lot of the modulation we see in the fMRI signal together with vigilance may be a combination of both some neural activity and as well as some of the autonomic effects that correlate with vigilance. And so to summarize this part, we are finding, we and others are finding that, there are fluctuations in alertness that relate to fMRI signals and connectivity, and that the reliability of these patterns may enable us to estimate someone's moment-to-moment vigilance changes with fMRI and perhaps capture some degree of task-related variability. Um, and as I mentioned, one reason that we're hoping this may be useful is that many fMRI datasets don't have EEG recorded together. It's pretty cumbersome to do the setup and recordings. And so perhaps this can enable incorporating information about vigilance into analyses where that's not available. But there's also some limitations to the work I've shown, which opens up future directions for this. And specifically, that measure I had shown mainly captures the relative change in vigilance across within a scan or within a person. And we also noticed that it doesn't yet capture a full range of vigilance fluctuations. And so we are also continuing this work to take other machine learning approaches like deep learning methods in order to try to better capture a more quantitative measure of alertness. So as we just discussed, I think that work on vigilance and fMRI has been showing us that not only is the brain itself engaged, but also so are the connections between the brain and the body. And vigilance is closely intertwined with these autonomic effects, through circuits like linking vigilance to changes in heart rate, breathing, and vascular tone. And one nice illustration of this phenomenon is in this recent study from Michael Chi's group. And here what they found was that if you look at signals that happen around microsleeps or brief periods of falling asleep, you see reliable changes in respiration and cardiac responses that come together with thalamocortical bolt responses. And you see that, both at the sleep onset as well as waking up, that there are these changes in both the physiology as well as the neural signals. And so another thing that we and others have observed that happens to our physiology as we're drifting between, during slow drifts in someone's vigilance, like falling asleep, is that there are also changes in the characteristics of physiological waveforms. So we can see like heart rate decrease, sometimes the heart rate variability increases, and in the scan, the respiration volume also started to show increased variability. These things are perhaps better known in the physiology literature, but coming at it from the fMRI kind of data-driven side, it was just interesting to see how these signals start to change across these different vigilance levels. Um, and so fMRI signals also change their characteristics throughout slow changes in vigilance. So here I'm talking not about these transient vigilance changes like in the previous study, but more like if we look over kind of like a couple of minutes of what happens as the person's maybe getting more and more drowsy. And what Van found here is that if you look at like the global signal, for example, the variation in global signal is higher when people are more drowsy than when they're more alert. If we use EEG as a measure of alertness again. And so relatedly, we also end up observing that the correlations between fMRI and peripheral autonomic signals, so these specifically low frequency variations in heart rate and respiratory volume, also seem to become more coherent as people are falling asleep, perhaps because of these changes in the characteristics of these signals as well. And so we quantified that a little bit further. So we looked at sliding windows of data. In each window, we estimated a baseline kind of level of vigilance based on the EEG ratio of alpha theta power. And for each window, we took the fMRI data and we asked to what extent low frequency physiology changes correlate or explain variance in the fMRI data across windows. And we generally find these negative relationships with, higher variance explained by physiology in lower arousal states. And so this is just expanding that analysis out to a number of different networks. We saw some of the most reliable effects in default mode network, for example, and also in CSF and white matter. So beyond the specific connections of low frequency physiology and vigilance, these systemic physiological effects are also important to consider, I'd say, in fMRI studies more generally, since they do come with effects in our bold signals. So if you were to take a voluntary deep breath in the scanner, you would then see an increase in your bold signal across many regions of the brain that can be even higher than the changes elicited by neural activity. And it's related to breathing, modulating cerebral blood flow and blood oxygenation. So this was a early study that where Rasmus Byrne had shown that not only when you take deep breaths do you see these correlations between low frequency physiology and fMRI, but also just in the subtle fluctuations in breathing depth and volume that seem to happen across the scan. One, I guess, kind of tricky thing in resting state data, and particularly when we don't maybe have measures of physiology available, is that it can often be hard to separate these kinds of effects from fMRI data in a data-driven fashion. So traditional spatial ICA-based methods seem to be better at isolating certain physiological effects in our data, like pulsations of vessels at each heartbeat, inflow effects, and the changes in magnetic field that are driven, by changes in the respiration. But these low frequency physiological responses are somewhat trickier because they do closely resemble bold signals. They rise from modulation of blood oxygenation, and they are also quite spatially widespread. And so you can often get a lot of residual effects in our data, even after some rigorous processing. And if we make, this is not, by the way, like this is not representative of like all data sets, but like, for particular subjects, you can indeed find that there are some subjects in which the correlation structure of the data seem to have effects that at least correlate with respiration and heart rate. And so one of my interests has been in this effects. Actually, I got quite interested in how we can model and characterize what's happening in fMRI data as we see changes in breathing and heart rate. And typically, a belt around the abdomen and a pulse ox are used to monitor these kinds of peripheral fluctuations. And so one way that we have been relating these kinds of signals to fMRI changes is through transfer functions. And so if you were to measure like a person's heart rate over time and convolve it with filters that look potentially like these kinds of shapes, you can estimate a component of the bold response that may be at least co-varying with that change in heart rate. And so, this could be, recording and modeling these physiological effects. We're not saying all of this is noise at all, but really that monitoring and modeling these waveforms can allow for studying many interesting things as well, such as how brain vasculature, brain-body interactions may change in health, also in certain disease states. And also, kind of going back to these close relationships between physiological signals and the brain states of a person, we also might be able to use these for indicating brain states that a person may be in. So to study this a little bit earlier, when I first started to look at a measure called heart rate variability, one of the things that we looked at was if we were to analyze like how heart rate variability changes over time, how would that impact the correlation of different networks, and particularly those that may be involved in salience or emotion regulation. And we found kind of that you could see these changes in connectivity throughout different windows of heart rate variability if we take that to be somewhat of a state related measure. And so, and we also, it's kind of tricky to study these physiological states because there may be neural and non-neural effects of these systemic signals, but we also try to like regress out changes in respiration, et cetera, in order to be able to try and isolate as best as possible some of the connectivity changes that were happening here. So heart rate variability has been used throughout literature to indicate a parasympathetic nervous system state, and is also known to be altered in major depressive disorder and other types of neuropsychiatric disorders. So potentially it could be interesting to relate some of these physiological indices to subjective or mood states in affective disorders, for example. But unfortunately, it's often the case that these physiological measures are either not collected during a particular scan, or they may be too noisy to be used. And so if we sometimes might see strange things happening in our recordings or in recordings of data that we've downloaded from other data sets. And so one idea that we and others have started to pursue is this idea of whether we can use fMRI data to extract what these physiological measures might be throughout the scan, and the components of those physiological effects in fMRI data if we don't have measurements available. And so there's been work by a number of different groups that have looked at reconstructing certain features of physiological waveforms, such as the timing of different heartbeats, or the timing of different the respiratory, the timing of each breath. And here we were specifically interested in low frequency variations in respiratory volume as well as heart rate. So, so this was an initial attempt at this, where we again have this goal of taking fMRI data as input and trying to output a continuous prediction of respiration volume across the scan that would match as best as possible what we might get if we are able to record that data over the scan. And in this early study, this earlier study, we focused specifically on respiration volume changes. And we compared two different models. So we looked at a convolutional neural network as kind of our more complex model, and we also looked at a simple single unit model, no nonlinearities. So it's just a linear network. And this set of models is operating in a sliding window fashion. So if you imagine taking a window of input data that consists of your fMRI signals sampled from a set of different brain regions, and kind of sliding that over, and at each window trying to predict the center point there in a continuous fashion. So this work was led by Jorge and Rosa in our group, and these were various single subject examples showing the actual versus predicted changes in low frequency respiratory volume over the scan. And this is just sample various degrees of accuracy. So this was a kind of good performing one, and then we get to kind of like medium, lowish performing ones. And then this is an example of if we were to use the measured versus the predicted respiratory waveforms in order to map that across the brain and understand which fMRI, which brain regions for which the fMRI signal was correlated with that waveform, we can get them to match pretty well. So across, those are single subject examples, but across the group, this is sort of how the performance looked. Our metric of performance was based on the temporal similarity, Pearson correlation between the estimated and predicted, or estimated and measured waveforms. And we compared, as I mentioned, these different models, and interestingly, there didn't seem to be much of a difference in the performance between those. So a much simpler model did just as well as this convolutional neural network for this problem. And when we take the weights of the single unit network, we can also plot them to try to understand a little bit the kind of spatiotemporal dynamics that are being used within that prediction window to try to and across brain regions to try to do this kind of decoding. But our median performance levels were not, as high as we wanted them to be. And so the direction that we next took was to try to build more complicated models to try to see, how much further we can try to boost that performance. And so Rosa in this work had started to use a LSTM network and particularly a multitask LSTM model that could predict respiration as well as heart rate together. So starting to extend it to heart rate as well. And we wanted to put those in the same model because we do know that there is shared information between respiratory and heart rate signals. So we might be able to leverage that. And we also expanded our data set. So we had 100 and something subjects before where we had made sure that the physiological data were of high quality before we started that. But here we used a larger pool of subjects, and these groups had kindly released some lists of subjects that they had determined to have good physical quality. So if we look at resting state data, this was able to boost the performance quite a bit on average compared to the previous model. And so now the median correlations were closer to 0.7 for the resting state data. And this is a kind of a good example of the correspondence between the measured and predicted respiratory volume and heart rate fluctuations over the scan. And we also started to try and extend this model to task fMRI data as well. And so if, just as a start, if we take the model that was trained on resting state data and apply it directly to the data from the HCP data of people that were performing different tasks with no further retraining or fine tuning, then we also see that the performance is decent, but not, as high as in resting state. So there is still room to try to improve that. And she has since been starting to now, also try to train on combinations of different tasks and use that to try and understand and see if we can increase the performance more. So just as a last slide, because I know we're close to getting out of time, as one illustration of the, if we were interested in, I mean, I mentioned there are potentially several uses of these signals, but if we were interested in something like denoising, then this is just an illustration of what happens to like default mode network, before and after removing a component of the physiological data that was linked with our measured physiological signals versus the predicted physiological signals. And this, some of the highlights here that it expands the anti-correlations that become visible in these data compared to if we don't regress out physiological signals. So just like our work on vigilance, we were hoping that these techniques for decoding the low frequency physiology can also help to enrich many fMRI data sets that may not have these signals available. So in summary, fMRI signals are very rich in terms of both their spatial patterns and their dynamics. We can extract information about vigilance states and physiology from fMRI alone. And again, emphasizing not only as a confound, but also as potentially valuable information about an individual and potentially about disease states. And finally, if we study these physiological states and incorporate them into our analyses, then they may lead to improved understanding of fMRI signals and new imaging markers as well. And so I'd just like to thank many people in our research group and our many collaborators on this work. And yeah, thanks again for your attention.
Mm-hmm. Thanks very much, Katie. It was great. Let me see if folks have questions for you. I certainly could chat with you a lot about various things, but I'd love to hear from others who are attending. There was one question, I think, from Brad, who was asking about when you were talking about the modeling of the heart rate, or no, about the respiratory volume.
Mm-hmm.
He was asking if the output was from that you were showing was from the where is it? I think it was from the deep learning model versus the simpler model.
Oh,
I think you kind of answered that, which was that they were about the same, basically, right?
Right. So in this first study where we compared the CNN to
Right there,
the
yeah.
linear model, right? Yes. So yeah, they actually gave pretty comparable performance. So yeah, thanks for the question. I'm not sure if I'm able to see. Oh, here we go. I found the chat window.
Oh, okay,
Great.
great. And maybe why don't you go ahead and unshare your slides? That way we can
Sure.
see
Let
more
me
fully. But see if the other.
stop sharing. There we go.
One question that came up was, how is vigilance index measured from fMRI?
Oh yeah. So that's so we try to learn a signature of vigilance from fMRI based on concurrent EEG at first. And so since EEG has more established signatures of vigilance, we at every TR we took the alpha power, the ratio of the alpha power and the theta power as a measure of that of an EEG vigilance, and then we get a time course across the scan of what that looks like. We then temporally correlate that with the fMRI data to see which regions change or go up and down as a function of what that EEG vigilance is doing, and that pattern is then used as the basis for deriving the fMRI vigilance measure. And that is more of then a spatial projection. This is similar to dual regression in a sense, if you are familiar with that method. But if you just like project this pattern back onto every scan to assess like to what degree that pattern is sort of expressed in each volume, then that was how we derived at every TR an estimated level of vigilance.
One question I had for you was, I thought this comes up a lot, which is you mentioned in your sort of introductory slides about how dynamics can sort of be a confound, right, in some ways. And so the way I've thought about that is the brain is very dynamic, of course, but So what we really need are models that can capture that variability, but then summarize it in some way that is stable, right?
Yeah,
So
completely.
I don't
Yeah.
know what is that. I just wanted to get your thoughts on
Oh yeah, yeah,
thinking
exactly.
about that.
Yeah, so I mean, that's right. So I should probably be clear on what I meant there too, because I because indeed, like if what I meant was really like if things were changing and we didn't model that, it
Mhm.
could look like unexplained variability.
Yeah.
Yeah, but actually the power, as like as your work has shown, as you know, if we are able to mine those patterns, and then that's a whole new level of what we are seeing in the data in terms of these, there may be, um, uh, these, there may be stable aspects of how these dynamics are expressed in an individual, like the dwell time of a connectivity pattern or a co-activation pattern, which may be a metric of that person's brain dynamics. And so then in that sense, that itself may be reproducible over, a long period of time in a person. So maybe like you scan them one day, you scan them another day, and you can see those being stable measures that may be unique to a person. So yeah, thanks for letting me clarify that as well.
Yeah, no, that's good. I think it's a very interesting question. I mean, I've seen some studies that tried to look at test retest and stability and things like that. And so if you're just looking at things that are stable, you're looking at modeling if dynamics and you do test retest, well then the state's probably different, right? And so the depending on what you're looking at, you might get low test retest reliability, but that doesn't necessarily mean that the information isn't useful and meaningful and could be summarized in some way.
Yeah, absolutely. Yeah,
Yeah.
totally agree.
Okay, well, I've got a bunch of other questions which I won't ask right now. I will instead, Cyrus, I think you were asking if you could be unmuted. So maybe, can you unmute yourself? Give it a try. If not, I can unmute you. Can't. Okay, hold on a second. Let me go ahead and I will unleash Cyrus here. Cyrus. Unmute. Go ahead, Cyrus.
Okay, can you hear me?
Yes.
Great, thank you so much. Great talk, very interesting. And yeah, this is what one dreams about, to collect all this breathing and heart rates, and it's very difficult. Anyway, so I think I had a couple of questions, but one was that you seem to have different response from the sympathetic nervous system to the fMRI response. That you could kind of pick up you could see the fMRI response on some subjects, and some you could see less the response of. And I was wondering, is this because of that the subjects that you had difficulty to pick this up from, did they have motion, or did they have another problem going on with their fMRI,
I see.
so that you would get a less good fMRI from that subject?
Okay, so just to make sure I understand the particular result you're referring to, is this where we were trying to use the fMRI data to estimate what the heart rate and respiration changes were across time, and the performance was different? Like some subjects we did really well, and some subjects it was less good. Is that the result you're referring to?
Well, I did see that, but I actually, the question arose earlier when you talked about the sympathetic nervous system, but I guess it's all the same, isn't it?
Oh yeah, I was trying to to remember that I didn't remember where I had seen where I had shown the
But you
variability
did show
response.
those lines, and you said that this was a great subject, and this is from two subjects that are average. And then I
Oh yes,
assume
yeah,
there are probably some subjects
sure.
that are less than average.
Sure. Yeah, yeah, oh, yeah, yeah, that's right. Yeah, they just look like they didn't really correlate. So, yeah, so That is true. We did find variability in our in how well we could do that, and I have a couple of thoughts on that. One is it we have done a little bit of further digging into that, and we find that as may kind of make intuitive sense, if there is more of respiratory or cardiac variation in the scan, then that somehow can be kind of better predicted by fMRI, because I think there's more an effective
Yeah,
fMRI than leveraging to predict it. So if someone has a very like kind of very stable heart rate and then they just have like a little bump or two, and we're using correlation to compare that relatively flat waveform with what we predict, then we're not going to see as great of a match. The other thing that we're wondering about,
mm,
which we are also testing further, is whether, so one tricky thing about using these physiological signals, both for training and evaluation, is that even though we tried to vet this to some extent and try to get the scans that had high quality physiological recordings, nonetheless, there are some artifacts and that we didn't completely denoise. Like, so we couldn't sit and fix like all the 400 subjects data. So we actually just left some of those in. Um, and
mm,
so when we do the evaluation, then that could come to hurt us then a little bit, because if we are actually like in the best case, if we are actually like what we're predicting is really good, but our ground truth is or what we're treating as ground truth is actually a little bit noisy, then that can manifest in what looks like a reduced performance. So we are actually doing further experiments now to see if we can use our method to kind of like fix noisy physiological waveforms in addition to kind of like, yeah, doing this learning. So that is, those are a couple directions. And I guess, of course, it's possible that, one model may not generalize perfectly to everybody. There may be some things about the physiology of that person's brain that is just not as consistent with maybe the other subjects. And so maybe there's just, we need to use more data, try to better learn the structure of individual variability better. So.
so as I get it, that it depends a lot on perhaps on the signal from the sympathetic nervous system, the heart rate and the breathing. So may I ask one more short question?
Last one, Cyrus. Then I'm going to put you back in your cage. No, just kidding.
Yeah. Uh, so you have done a lot of measurements with the heart rate, with the breath, and I saw the pulse oximetry, the pulse, this thing that you measure for COVID on the finger too. So there's a lot of problems that these gadgets don't work and stuff. And I was wondering, do you have any feedback on what is most valuable to collect? Is it okay to just stick on a pulse oximetry thing on the finger? Is that the best? Or what is your what do you think?
Yeah, I think the pulse oximetry is very valuable, but indeed it can be tricky to get it to, like, if it doesn't stick on the subject quite right and they move their hand a little bit, then the whole recording is gone. Um, or if they, yeah, are just, yeah, sometimes it's just not quite attached correctly. And so I think but I think that signal, we've so far been able to get what seems to be a lot of valuable information from it, because we have been looking not only at the timing of the heartbeats, which you can see as the pulsations in there, but also these slower variations in amplitude, which seem to track the sympathetic nervous system response and relate to the kind of blood flow or blood volume. And so that is one, yeah, that I would agree that you can derive quite a bit of useful information from. And then the respiration. I think that there are fancier, but maybe more difficult to obtain or record, like, so end tidal CO2, for example, is maybe a little bit more desirable or complementary to have than to have to the respiration belt, because the respiration belt, and when we are deriving this like low frequency changes in respiratory volume, it's kind of trying to approximate like a tidal volume measures. But then again, there's a trade-off, because to set up a system that monitors that like may be a little more difficult. And
Yeah,
so I think that for routine scans, since the pulse ox and respiration belts are so widely available, then I think we can get a ton of useful information from those measures if we try to collect them.
Hmm.
thank you.
Great. So we're right at 12. So I think we'll go ahead and close out the talk now, but we do have the questions. I've got some questions here that I will save, and we'll follow up with a Slack channel so that we can have follow-up discussions. I think there'll be some good discussions there. And also the video will be shared there as well in various places. So thanks again, Katie, for a wonderful talk and for your time with us.
Thanks so much.
And we look forward to continuing the discussion.
Yeah, thanks so much for hosting me. I really
Yeah.
appreciate it. Yeah,
Great.
great to see you.
Okay.
Alrighty.
Thanks everyone for joining. Okay. Have a great weekend, everybody. Bye-bye.